Hello student, welcome back to my YouTube channel. This is Dr. Sumit. Today tutorial I will discuss about nucleic acid. Nucleic acid, you will learn about nucleic acid in second lesson, that is chemical basis of life, and also this knowledge is very important in molecular biology lesson. So let's begin. Nucleic acid. Nucleotide is the monomer of the nucleic acid. So you have huge number of nucleotide, you call them polynucleotide. Polynucleotide form the polymer, that is nucleic acid. And nucleic acid contain of carbon, hydrogen, oxygen, nitrogen and phosphorus. Because of the high molecular weight of the nucleic acid, you call them a macromolecule. When you consider about nucleotide, basically based on nucleotide, there are two type of nucleic acid, DNA and RNA. DNA is deoxyribonucleic acid and RNA is ribose nucleic acid. When you take the chemical composition of nucleotide, nucleotide composition compared to the 5 carbon sugar backbone, that is you call pentose sugar and nitrogen base, and the other one is the phosphate group. Phosphate group is the group that giving acid nature of the nucleic acid, that is why you call it a nucleic acid. When we take nucleotide, nucleotide composed of nitrogen base, pentose sugar and a phosphate group. If you remove the phosphate group from the nucleotide, it is, you call it as a nucleoside. When we take pentose sugar, chemical structure of the pentose sugar with carbon 5 sugar. In the RNA, it is ribose pentose sugar. When you take ribose and deoxyribose pentose sugar, what is the main structural difference is the second place hydrogen in the deoxyribose, but it is hydroxyl in the ribose. So you have more one oxygen atom in ribose, so you call it the deoxyribose. Less oxygen, one oxygen less in the deoxyribose. When you talk about nitrogen base, there are basically 5 nitrogen bases, and you can categorize them into purine and pyrimidine basically based on their size. Purine, there are two types of purine, adenine and guanine. They have large two rings. And when you take the pyrimidine, there are 3: thymidine, cytosine and uracil. And they have only one single ring. And when you consider thymine, you can only see in thymine in DNA, and uracil only can be seen in RNA. But cytosine can be seen in both RNA and DNA. When we take DNA formation, nucleotide, you take the 5 end 5 end of the pentose sugar bind with phosphate group, and third carbon bind to the next nucleotide phosphate group. Like this, sugar and phosphate will form the structure, and opposite is happening in the other side. It is 3 end here and 5 end here. And when you talk about the nitrogen bases, adenine coupled with thymine with 2 hydrogen bonds, and guanine coupled with cytosine with 3 hydrogen bonds. So when you take DNA and RNA, they are capable of their own replication. That means they can copy their own, and these are found as genetic material in. So DNA basically the genetic material, and some viruses you can see RNA as the genetic material. Example is influenza virus. The basic function of the DNA and RNA is the store and express hereditary information, and this information transfers from one generation to the next generation. When we take the structure of the DNA, there's a proposed structure that is called DNA double helix model, that is proposed by Watson and Crick. So you call them a Watson-Crick model. James Watson and Francis Crick. They have proposed this one with the evidence of crystallographic figures that is done by Rosalind Franklin. When you take a DNA molecule, it's like a twisted ladder. The backbone of the ladder made up of phosphate and sugar molecule, and steps are made up of paired nitrogen bases. So the two parallel strands are there, and these two are opposite. 5 prime to 3 prime in one side and 3 prime to 5 prime in the other side. So you call them, these two chains are complementary and these two chains are anti-parallel. When you take complete one turn, you can have 10 base pairs in that one turn. Purine bases with pyrimidine, that is adenine go with thymine with 2 hydrogen bonds, and guanine go with cytosine with 3 hydrogen bond, and adenine also go with uracil if it is in RNA molecule. When you consider about RNA is single strand, and you can see ribonucleotides, and there are three types of RNA: mRNA, tRNA, and rRNA. mRNA. mRNA is the messenger RNA, and these are like messengers. So they are copy the genetic information, and they are transport for genetic information from nucleus to the cytoplasm. If you take tRNA, tRNA is the trans RNA. The function of the tRNA is to transport amino acid to the site of protein synthesis. And rRNA, that is ribosomal RNA. Basically, ribosome is formed with rRNA. So they are provide the sites to resemble polypeptide chain. And when you take DNA and RNA, DNA is double stranded and RNA is single stranded. DNA composed with deoxyribonucleic acid, RNA composed of ribose nucleic acid. Those are the differences between DNA and RNA. And also in functional situation, DNA has the function in genetic information, and some viruses it is RNA. Thank you. In this tutorial, I have discussed about nucleic acid. Dr. Sumedu from BioSir YouTube channel. If you haven't subscribed my channel, subscribe for more biology lessons. And also you can visit my website mybiotutor.com. Thank you.